Good afternoon, dear students. We are now going to study the collection and handling of water sample. See, in the last video, I explained you why we need to perform bacteriological analysis of water. Now, when you want to perform the bacteriological analysis of water, you have to go to the source of water, collect the water sample, and bring it to your laboratory. Okay, so we need to understand how to collect the water and how to handle the water, how to bring the water from the site to the laboratory. So, we are going to study the collection and handling of water samples. Now, the principal objective of sampling water, collecting water sample for bacteriological examination are: number one, the sample should be a representative of the source or the supply. Okay, the sample which you bring to your laboratory should represent the source or the supply from where you have collected the water. And the second objective is to preserve the original bacterial count of the collected water sample. When you collect the water from the source, there is some some number of microbes or bacteria are present in the water. You have to preserve the same count till you perform testing of the water. So the sample should represent the source of the water supply, and you have to preserve the bacterial content of the collected sample. These are the two principal objectives, and hence we have to study how to collect and handle the water. The first and most important is the sample bottles. Okay, what care you should take, or what kind of sample bottles you are supposed to use. Now, a sample of water for bacteriological examination are to be collected in a clean, sterile, glass stoppered bottles of good quality. Okay, the glass bottle should be clean, number one, sterile and stoppered. Okay, generally wide mouth bottles of 100 ml capacity are preferred for collecting water sample. Right, so the bottle should be clean, sterile and stoppered, made up of glass, having wide mouth and a capacity of generally 100 ml is preferred. How to clean the bottle? The bottle is thoroughly cleaned before using. It is cleaned with sulfuric acid and potassium dichromate. Then it is rinsed with clean water. It is dried by draining the water, and it is sterilized in a hot air oven, that is by dry heat at 170 degrees Celsius for at least 1 hour. Or you can also sterilize the bottle by autoclaving at 15 pounds pressure for 20 minutes. We have to take care that the neck of the bottle and the cap are protected. The stopper, the cap of the bottle, that is stopper of the bottle, and the neck of the bottle have to be protected from getting contaminated. And how can we do that? The neck and the stopper of the bottle are wrapped with brown paper and then sterilized in the autoclave. So after sterilization, we don't have to touch the neck of the bottle. Now, if you are going to collect chlorinated water, if the water is treated and we are going to collect chlorinated water, then for collection of chlorinated water, 0.02 to 0.05 grams of sodium thiosulfate is first placed in the sampling bottle, and then the bottle along with sodium thiosulfate is sterilized by dry heat. Okay, when we are collecting chlorinated water samples, treated water samples which contain chlorine, we have to place 0.02 to 0.05 grams of sodium thiosulfate in the bottle, then sterilize the bottle, and then collect the water sample. That means the chlorinated water sample is collected in a bottle which contains sodium thiosulfate. Why we need to do this? Okay, because this precaution is followed to preserve the original bacterial count. What will happen? If the chlorine present in water will slowly reduce the bacterial count. You collect the sample, bring it to the laboratory and analyze. There is some time lag, a time gap between the you can say time at which you collect the water sample and perform the analysis. So during the time gap between the two, the microbial content or the bacterial count may reduce because of the residual chlorine that is present in the water. So to preserve the original bacterial count, the dechlorinating agent is placed in the sampling bottle. If the dechlorinating agent is not used, then chlorine that is present in the water sample will reduce the number of living bacteria in the sample, viable bacteria in the sample before examination. So we have to follow this precaution to maintain the original bacterial count. Okay, now how to collect the representative samples? Okay, so collection of representative samples. We have to take great care to collect fair samples and also to protect it from any type of contamination that may change its bacterial content. Okay, so we have to take care to collect fair samples, number one, and protect the original bacterial count. We should not allow any type of contamination which can alter the bacterial count. Okay, so number one, collection of tap water samples. If the sample is to be collected from the tap, then water should be allowed to flow for at least 5 minutes. If you're collecting water from the tap, just hold the bottle in your right hand and the cap in your left hand and collect the water. Before you start collecting the water in the bottle, allow the water to flow for 5 minutes to wash away any kind of contaminating microorganisms that are likely to be present at the faucet. Okay, some kind of contamination or dirt or microbes may be present at the opening of the tap. So we want to remove all those microbes or dirt which may be present at the opening, that is the faucet, and that is why we allow 5 minutes, allow the water to flow for 5 minutes and then collect the water sample. So it's very simple to collect water from tap. Next, number two, how to collect well water samples. Now, if the well is provided with a pump, and of course, if it is provided with a pump, there is a tap. So, similar precautions are observed as in case of collection of tap water. If the well is not provided with a pump, the sample is to be collected with the help of a clean, sterile bucket. Okay, we have to collect sample with the help of a clean, sterile bucket. But nowadays, this is not the case. Certain type of water samplers are available nowadays. Specific water samplers are available to collect samples from deep well water. Okay, so one of the samplers is Gobel J6. Okay, so these samplers are to be plunged into the water, into the deep water, and collect the water sample. And if you don't have a specific water sampler, then we have to use a clean and sterile bucket to collect the water. Now, if you are collecting stored water or surface water, like from a river, from a lake, how to do that? So, stored and surface water sample collection. When we are sampling surface waters, we have to take particular care, or we have to follow certain instructions to prevent contamination from your finger. Okay, when you are collecting the water, there are chances that the water may get contaminated because of your fingers. So, we have to take certain care. Number one, if we are collecting standstill water, the cap of the bottle is removed with the left hand. Okay, and then the bottle is plunged with the mouth downwards into the water to a depth of 30 centimeter, and then the bottle is inverted. Okay, when the bottle is almost filled, it is removed out of the water and stoppered. Okay, listen again. If you are sampling still waters, standstill waters, which are not moving, okay, if you are collecting standstill waters, cap of the bottle is removed with the help of the left hand. The bottle is then plunged into the water downwards to a depth of about 30 centimeter and then inverted. And the bottle starts filling with water now. When it is almost filled, remove the bottle and stopper it. If any kind of current is present in the water, if any current exists in the water, the mouth of the bottle is directed against the current to avoid contamination. If you collect sample where there is water current, then naturally it will come with you can say force and may get contaminated because your fingers. So, if there is any kind of current existing in the water sample, then in the water body, then the mouth of the bottle should be directed against the current to avoid contamination. Okay. Next thing, for extensive stream studies, when we have to detect the source and extent of pollution, when you are performing extensive stream studies, like you're not just detecting whether what kind of bacteria is present, you're also checking the source of contamination, the extent of pollution. We have to take many representative samples from different sites. Okay, from the same water body, we go on collecting water from different sites. The number of sampling site depend upon the number of sources polluting the stream. See, if there are 4 or 5 industries around the stream, then from all the 5 industries, wastewater will be discharged into that stream, into the water body. So, we have to first locate what is the site at which the water body is getting polluted, or what is the site of where the stream is getting polluted, and then we have to collect the sample from each site. Okay, so from different sites we collect the water sample, and generally water sample is collected during peak hours of pollution. When there are peak hours of pollution or the waste is getting dumped into the stream, go and collect the water from different sites where pollutants, or you can say wastewater, is being added to the stream. Now, if you want to estimate what is the average pollution, you have to take sample from a point where there is complete mixing of polluted water. Okay, so if you just want to estimate the average pollution in a water body, then we have to collect a single sample from a point where all the pollutants get mixed into the water. Okay, now in case of rivers, the sample has to be collected from different depth. Okay, see, if you're collecting river water, you have to collect the sample from different depth, right? And all these samples should be mixed to get a composite sample, and then you perform bacteriological analysis. If you have to collect the sample by using a boat, or if you have to get inside the water body, okay, you have to go inside the water body, and then in the middle of the water body you need to go sometimes and then collect the water. In that case, we have to go by a boat or a steamer. Okay, so when you are to be taken by using, when the sample is to be taken by using a boat or a steamer, collect the sample from upstream site from the boat. Okay, from the upstream, not from the downstream. If it is not, okay, sometimes collecting water from upstream site using a bottle is not easy, right? Because the bottle will not plunge into the water. So if it is not possible to collect the sample from upstream site, attach some weight to the at the base of the bottle, attach some weight at the base of the bottle, and then collect the sample. It can be easily collected, right? So once your collection is over, you have to perform the analysis. And before performing analysis, you have to come bacteriological analysis, and you have to come back to your laboratory. So you have to store the samples. How to store the samples? If the samples are to be examined immediately after it has been collected, it is to be stored in its original state. Okay? Okay, if the sample cannot be examined immediately after it has been collected, it has to be stored in its original state to maintain the bacterial count. It has been observed that bacterial count may increase or decrease. How does it increase? Due to multiplication of typical water bacteria. The bacterial count may increase because of multiplication of typical water bacteria. Okay. Further, temperature influences the bacterial count. How? The organisms from intestinal tract will tend to die at elevated temperature. If the temperature is higher, microbes from intestinal tract may die. This may reduce the bacterial count. Okay, so because of multiplication, bacterial count may increase, and if the temperature is higher, the intestinal tract bacteria number will reduce. So during storage, the bacterial count may increase or decrease. So we have to properly store the sample and bring them to the laboratory. To prevent such complication, we have to follow certain precautions. What are those precautions? Number 1, all samples should be examined as early as possible. Number 2, if the sample has to be examined within 1 hour after collection, it has to be iced. The sample should be held below 10 degrees Celsius. during a transport period of 6 hours, maximum transport period of 6 hours. On arrival in the laboratory, the sample should be processed within 2 hours. And in no case, the examination should delay more than 30 hours. Okay. So you have to examine all the samples as early as possible. Number one, precaution. Number two, if you are not going to examine it within 1 hour, ice the sample, convert it into ice. The sample during a maximum transport period of 6 hours, you have to store the sample below 10 degrees Celsius, and upon arrival into the laboratory within 2 hours, perform the examination. And in no case, examination should delay more than 30 hours. Okay, so these precautions are to be followed, and once you bring the sample to the laboratory, perform bacteriological analysis. Right? So that is all about collection and handling of water sample.